Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. I'm Dr. Loai and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. Alright, in our previous lectures, we completed the first part of molecular biology ASCP exams focusing on the molecular science sections, alright, which account about 20 to 25% of the total exams. Today, we'll move to the second sections of the exams, molecular techniques. These sections including several topics, as we see, such as the nucleic acid isolation, RNA DNA manipulation, separation and detection, nucleic acid amplification sequencing, and other techniques, which together make up 30 to 35% of the total molecular biology ASCP exams. So let's focus on the question related to the first part of molecular techniques, nucleic acid isolations, and review molecular biology ASCP questions from the previous exams. Let's go. All right, the first question we have today. Which chemical is commonly used in liquid phase manual nucleic acid extraction? 15 seconds. All right, the correct answer is phenol chloroform. Phenol chloroform extraction method is widely used techniques for purifying DNA or RNA. The process begins with the cell lysis, where the cells are broken open to release their content, including DNA and RNA. Next, the sample is mixed with phenol and chloroform and then centrifuged. So this creating two distinct layers. The upper water-based layer containing the purified nucleic acids and the lower layer which traps protein and other unwanted material. Okay, so finally the nucleic acid containing the upper water-based layer is carefully extracted, resulting in clean samples of DNA or RNA free from protein and other contaminants. All right, let's move on. All right, the next question we have here. What is the advantage of using magnetic beads in a solid support manual nucleic acid extractions? All right, the correct answer is absence of forceful column washing. Actually, magnetic beads based nucleic acid extractions offers a significant advantage over traditional column-based methods by eliminating the need for forceful column washing. All right, in column-based techniques, washing step often require high-speed centrifugation or vacuum pressure, which can lead to sample loss or sharing of long nucleic acids molecules. In contrast, magnetic beads allow for gentler process where nucleic acids bind to the beads and are separated using a magnet. So this process reduces the mechanical stress on the molecules, leading to higher quality extraction with improved yield and integrity. All right, let's move on. All right, next question. What is the primary advantage of using magnetic beads based automated nucleic acid extraction systems? The correct answer is high specificity and efficiency. All right, let's move on. All right, next question. In manual nucleic acid extractions, what is the purpose of using ethanol precipitation? The correct answer is to precipitate nucleic acids. Actually, ethanol precipitation is a widely used method for concentrating and purifying DNA or RNA. So, by adding ethanol and salt like sodium acetate to the nucleic acid solutions, the solubility of nucleic acid decreases, causing them to clump together and separated from the liquid. Then we can use centrifugation to collect the nucleic acid pellet while removing unwanted impurities. All right. So, and finally, the purified DNA or RNA can be washed out and dissolved in a clean buffer, making it ready for further experiments. All right. Okay, let's see the next question here. What is the significant benefit of using silica-based column in automated nucleic acid extraction? Alright, the correct answer is high purity and yield. Actually, silica-based columns are widely used in automated nucleic acid extraction because they provide high purity and yield of DNA and RNA. The silica membrane efficiently binds nucleic acid in the presence of chaotropic salts, allowing impurities such as the protein and lipids to be washed away. So this results in highly purified nucleic acid suitable for sensitive downstream application. Alright? Alright, next question. What is the purpose of using silica membrane in manual nucleic acid extractions? Alright, the correct answer is to bind nucleic acids. Actually, in manual nucleic acid extraction, a silica membrane is used to capture DNA or RNA. This works because the nucleic acid naturally stick to silica when achotropic salts are present. Alright? So, after the DNA or RNA sticks or bind to the silica membrane, any remaining contaminants such as protein, lipids, or salt are removed by washing the membrane with a special solution. Then, to collect the purified nucleic acids, a clean buffer or water is added, which release the DNA or RNA from the membrane. Okay, let's move on. All right, let's see in the next question. Which step is the crucial in the manual nucleic acid extraction process to ensure the removal of proteins? 15 seconds. Cell lysis, proteinase K digestion, ethanol washing, or DNA elutions. The correct answer is proteinase K digestion. Actually, in manual nucleic acid extractions, proteinase K digestion is important steps for removing proteins. This proteinase K we consider it as the strong enzyme that break down proteins and preventing them from interfering with the process like PCR or sequencing. So after these steps, I mean the digestion step, the samples undergoes further amplification such as the ethanol washing to remove any remaining contaminant. So this ensures that the final nucleic acid extract is the clean and ready for accurate analysis. All right? If you found this content helpful, don't forget to like, share, and subscribe. Your support means the world to me and help me create more valuable content for you. Thank you for being part of this journey. So let's continue and see the next question here. Which reagent is commonly used to lysis cell in manual nucleic acid extraction? The correct answer is SDS. SDS or sodium dodecyl sulfates is commonly used to lyse cells in manual nucleic acid extraction process. All right. So these reagents break down cell membranes and releasing nucleic acids for further use. All right. Let's move on. All right. Let's see the question here. What is the role of isopropanol in manual nucleic acid extraction? The correct answer is nucleic acid precipitation. Actually, isopropanol is used in manual nucleic acid extractions to precipitate DNA or RNA from the solutions. So, when mixed with the samples, isopropanol reduces the solubility of nucleic acids, causing them to aggregate and form a visible pellet upon centrifugation. So, these steps help concentrate the nucleic acids, making them easier to collect and resuspended in a suitable buffer for downstream application like PCR, sequencing, or electrophoresis. So, compared to ethanol, isopropanol require a lower volume to achieve precipitation and can be more efficient for recovering nucleic acids from dilute samples. Right? Let's move on. Alright, the next question here. Which of the following is the common step in both manual and automated nucleic acid extraction methods? The correct answer is centrifugation. Centrifugation is the common step in both manual and automated nucleic acid extraction methods, used to separate nucleic acid from the other cellular component. Alright, let's move on. Okay, let's see the next question here. What is the primary purpose of using lysis buffers in automated nucleic acid extractions? Alright, the correct answer is to lyse cells and release nucleic acids. Actually, these lysis buffers are essential in automated nucleic acid extraction because they lyse cells and release DNA or RNA. These buffers typically contain detergent to disrupt cell membranes, chiotropic salts to denature proteins and stabilize nucleic acids, and sometimes enzyme like proteinase K to digest protein. So, proper lysis ensure efficient nucleic acids recovery. All right, let's move on. All right, let's see the next question here. Which reagent is often used to remove RNA during DNA extraction in manual methods? Alright, the correct answer is RNase. Actually, during DNA extractions, RNase is commonly used to degrade RNA that may be present in the sample, right? So, since the RNA and DNA can coexist in the cell extract, RNase specifically breaks down RNA molecule without affecting DNA. This ensures that the pure DNA samples free from RNA contamination. Alright, let's move on. Alright, let's see the next question here. Which reagent is commonly used to inactivate RNAs during manual nucleic acid extraction? The correct answer is EDTA or EDITA. Actually, EDITA plays a crucial role in manual nucleic acid extraction by protecting RNA from degradation. It works by binding divalent metal ions like magnesium and calcium, which are essential for RNA's activities. All right? So, without these ions, RNAs cannot function, ensuring that the RNA remain intact during extraction. So, this is especially important when isolating RNA for application like RT-PCR and sequencing. Additionally, EDITA also protect DNA from nuclease. EDITA also protect DNA from nuclease and help stabilize nucleic acid in a solution. All right, let's move on. Okay, let's see the next question here. What is the purpose of using a chiotropic agent in mononucleic acid extraction? All right, the correct answer is to denature proteins and nucleic acids. Okay, so these chiotropic agents like guanidinium salts work by breaking down the structure of proteins and nucleic acids. During the cell lysis, they denature proteins and separate nucleic acid from the other cellular component. This is not only released for the DNA or RNA, but also inactivate enzymes like nuclease that could degrade the genetic materials. All right, let's move on. All right, let's see the next question here. Which step in manual nucleic acid extraction is the critical for removing contaminants such as the proteins and lipids? All right, the correct answer is washing with ethanol. All right. So during manual nucleic acid extractions, washing with ethanol is the key step in removing contaminants like protein and lipids. All right. After nucleic acid are precipitated, ethanol washing help eliminate residual salt, protein, and other impurities that may be interferes with the downstream applications such as the PCR or sequencing. Okay, let's move on. All right, let's see the last question we have today. To store RNA for longer than 6 months is recommended to. As we know, RNA is highly fragile and degrades quickly due to RNase and hydrolysis. To preserve its integrity, proper storage is the crucial. So let's break down how different temperature affect RNA stability. Let's start from option 1, minus 80 degree. This is the best option for long-term storage, especially when using specialized buffer containing RNase inhibitors and pH stabilizers. These help maintain RNA integrity for extended period. All right. Let's see the second options. Minus 20. Minus 20. While it slows down RNA degradation, it is not cold enough to fully prevent breakdown, especially if the RNA is stored in water rather than stabilizing buffer. For the option 3, 4 degree. 4 degree is suitable for short-term use, I mean few hours to days, but RNA will gradually degrade over time, making it unreliable for long storage. All right. For the last option, room temperature. Room temperature is extremely unstable, as the RNA can be degrade within hours. All right. So the correct answer here is keep it at minus 80 degree in a suitable RNA storage buffer. All right. That's all for today. I hope you enjoyed this lecture and look forward to hearing from you what you think of it. And also, please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. I also encourage everyone taking the test to share your questions with us so that everyone can benefit. That's all. Salam.